When we think about the discovery of DNA, our minds usually jump to one specific moment, one iconic image. But the story we've all been told, the one that kicks off in the 1950s, well, it's not the whole story. In fact, it's more of an ending than a beginning.
Right, this is the textbook version. It's the story you learn in school, the heroic moment when Watson and Crick revealed the double helix. And look, that discovery was absolutely monumental, no question about it. But if we start the story there, we miss almost 100 years of crucial and honestly pretty contentious science that came before it.
So let's get into it. The actual starting point is way less glamorous. We have to go back to 1869. And this story doesn't involve a race for a Nobel Prize. It involves a young Swiss doctor meticulously scraping pus from used surgical bandages to isolate a brand new substance from the nuclei of white blood cells. This is the moment the molecule we now call DNA was first actually found and described.
So here's our game plan for this explainer. We're going to tear down that popular origin story. We'll start with a quick biology refresher, then we'll rewind to the true beginning, the discovery of a substance called nuclein. After that, we'll get into the big fight between chemists and cytologists, untangle all the confusing names they were using, and finish up by looking at how we decide who gets remembered in science.
All right, before we can really get into the historical drama, we need to be on the same page about the molecule at the heart of it all. So let's do a quick rundown of the basics of our genetic code. So inside almost every single one of our cells, there's a nucleus. Think of it as the cell's command center. And inside that nucleus is where our genetic instruction manual, or DNA, is stored. It's not just floating around in there. It's neatly wound up and packed into these structures we call chromosomes. You got 23 pairs of them. Now, if you could unravel one of those chromosomes, you'd find the DNA molecule itself. And if you zoom in even further, you see it's built from a repeating unit called a nucleotide. Every single nucleotide has 3 parts: a phosphate, a sugar, and one of 4 nitrogen-containing bases. The phosphate and sugar link up over and over again to form a long backbone. And this is the brilliant part. The structure looks like a twisted ladder. The 2 long backbones are the sides of the ladder, and the rungs are made by the bases pairing up in a very specific way. Adenine, or A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. That's the rule, and it's what holds the whole double helix together. Okay, now that we have that modern picture in our minds, we can go back in time. Let's rewind the clock from 1953 all the way to 1869 to a lab in Germany and a 25-year-old Swiss biochemist named Friedrich Miescher. Miescher's goal was pretty simple. He wanted to study the proteins in white blood cells. And his source material? Well, it was those used bandages from a local clinic. But while he was trying to isolate the cell nuclei, he stumbled onto a substance with some really weird properties. It was loaded with phosphorus, totally different from the proteins he was looking for, and it wouldn't break down when he used protein-digesting enzymes. Since he found it in the nucleus, he gave it a straightforward name: nuclein. Now, Miescher was a chemist, through and through, and he knew that pus wasn't the ideal source material. He needed something cleaner, purer. He found it in salmon sperm from the Rhine River. See, these cells are basically just tightly packed nuclei, which meant he could get way more nuclein and really start to analyze its chemical makeup properly. Miescher knew right away he'd found a whole new class of molecule, something really fundamental. So why is his name basically unknown today? Well, a big part of the answer is a major clash that was happening in biology at the time, the fight between the chemists and the cytologists. This slide just lays it out perfectly. You have 2 different worlds looking at the exact same thing, the nucleus. On one side, you have Miescher, the chemist. His whole approach was extraction. Grind up the cells, break them apart, and figure out what stuff is inside. On the other side, you had guys like Walther Flemming, the cytologist. His method was staining. He used dyes to see how structures looked inside a cell. They were speaking completely different scientific languages. And the tension between these 2 camps was thick. From Miescher's point of view, just adding color to a cell and looking at it under a microscope told you nothing about what it was actually made of. He privately scoffed at the whole field, calling them a guild of dyers. He believed real understanding only came from chemical analysis. And Miescher's analysis was incredibly sophisticated for his time. He didn't just find nuclein in isolation. He correctly figured out that inside the cell, it was bound together with a protein which he also isolated and named protamine. He discovered what we now call a nucleoprotein complex.
This was decades ahead of its time, and it was a chemical reality that was completely invisible to the people just looking through microscopes. So, this huge divide in methods led to, you guessed it, a total mess when it came to naming things. The two groups weren't just using different tools, they started creating different vocabularies for the same stuff inside the nucleus. They were completely talking past each other. You can see the battle play out on this timeline. Miescher discovers nuclein in 1869. Then, years later, the cytologist Fleming sees this stuff in the nucleus that soaks up dye really well, and he calls it chromatin, which just means colored material. Finally, in 1889, one of Miescher's own former students, Richard Altman, manages to strip the protein off of nuclein, and he renames the pure stuff nucleic acid. Miescher's original name was getting buried. And it wasn't just the confusing terms. A whole storm of other factors was working against Miescher. His own boss was so skeptical that he held up the publication for 2 years to double-check the results. The Franco-Prussian War caused more delays and stirred up national rivalries. But maybe the biggest problem was that the beautiful pictures of chromosomes the cytologists were making were just more exciting and easier to understand for other scientists than Miescher's papers about some chemical gunk. So as a result, Miescher's work and his ideas about what nuclein might actually do were mostly ignored. But his legacy is so much more complicated and frankly more ironic than just being forgotten. Okay, this is just incredible. Get this. In a private letter he wrote in 1892, Miescher started speculating about what nuclein was for. He argued that because the molecule was so massive and complex, it could be arranged in countless different ways. He suggested this physical variety could be the basis for heredity, that it could store information, just like the 24 to 30 letters of the alphabet can be combined to make every word in every language. He was basically dreaming up the concept of a genetic code. And this brings us to the great irony of his entire story. Step 1, he invents the first ever method to get nucleic acid out of a cell. Step 2, his ideas about what it does are totally ignored. Step 3, his method, his chemical recipe is so good that it becomes a standard tool in labs for the next 50 years. And that leads to step 4. The very success of his method helped erase him from the story. There's a term for this in the history of science, actually. It's called black boxing. A technique becomes so reliable and routine that people just use it without thinking about how it works or where it came from. It's just a tool in a box. Miescher's protocol got black boxed. It became a standard lab procedure, completely disconnected from the man who invented it. Ultimately, what happened here is that history was written by the winners of a scientific turf war. Cytology and the field of genetics that grew out of it built an origin story that started with things you could see, like Fleming's chromosomes, not with Miescher's invisible chemical substance. The story was written to begin with the picture, not the stuff the picture was made of. And all of this leaves us with a really important question about how we remember science. Who gets the credit for a discovery? Is it the person who first finds a new substance? Or the person who finally figures out its grand purpose? The forgotten story of Friedrich Miescher shows us that the answer is almost never as simple as we think.